Hello students, welcome to EPG Pathshala. I am Dr. Nilofer from Deepas, Delhi. Today we are going to talk on module Structure of Nerves, Neurons, Blood-Brain Barrier, Generating Nerve Impulse. This is from the paper Membrane Biophysics. And the objective of the talks are structure of nerve, types of neurons, conduction of nerve signals, blood-brain barrier, and types of glia. Now coming to the first part, that is to introduce the topic. What is that? The nervous system is the system which coordinates voluntary and involuntary actions of the body and transmits signals to and from different parts of the body. In invertebrates, it consists of two main parts, the central nervous systems and the peripheral nervous system. The central nervous system includes the brain and the spinal cord, and the peripheral nervous system consists mainly of nerves. Nerves that carry signals from the brain are called motor or efferent nerves, while those nerves that send out information from the body to the central nervous systems are called sensory or afferent nerves. Most nerves serve both functions and are called mixed nerves. Now, what is neuron? The neuron is a common term for a nerve cell. Nerve commonly referred to a bundle of nerve fibers from different neurons. And neurons originate, processes, transmit and receive nerve impulses, and they are connected to each other or to the other cells in muscle, organs or in glands. Nerve impulses travel electrically along the neurons and transmitted by the chemical messengers, these are also called neurotransmitters, to the next neurons across a tiny gap. It is called a synapse between the neurons and the neighboring cells, which is known as a target cell. In addition to neurons, the nervous system contains large number of other cell types. These are known as neuroglia. And the function of the neuroglia are to protect, nourish and support neurons. Although neurons come in different shapes and sizes, they have common characteristics. Structure of nerve. What is neuron? A neuron is a nerve cell with all its processes. Neuron is the structural and functional unit of the nervous system. A typical neuron consists of a cell body or soma and two types of processes. These are axons and dendrites. In the figure below, you can see that the first part is the cell body or soma. In the middle is axon, and in the last there are axon terminals. They are different part of the nerve cells. So the parts of the nerve cell and their functions. The first one is dendrites, cell body, neuronal membrane, axons and nerve endings. Parts of the nerve cell and their functions. The first one is dendrites. Dendrites are the short cytoplasmic processes of the cell body, and these dendrites receive stimulus or impulse from the axons of the other neuron through synapse and conduct nerve impulses induced by stimuli towards the cell body. The dendrites at their origin from the cell body are 5 to 10 micrometer in thickness, but gradually their thickness decreases by profuse branching. They are also called receptive organs. Dendrites branch out in tree-like fashion and serve as the main apparatus for receiving signals from other nerve cells. The dendritic membrane under the synapse has many receptors that detect the neurotransmitters in the synaptic cleft. A nerve cell can have many dendrites which branch many times. Their surface is irregular and covered with dendritic spines, which are where the synaptic input connections are made. The cell body. The cell body or soma is nucleated cytoplasmic portion of a neuron. The size of each cell body, which varies from 4 to 100 micrometer in diameter, and it may be pyramidal, fusiform, pyriform or irregular satellite in shape. The cell body have a large spherical central nucleus along with the large number of nacelle bodies or granules. These are group of ribosomes used for protein synthesis within the cytoplasm. The cytoplasm of the cell body is also called nucleoplasm. It makes all the proteins for the other neuronal parts like dendrites, axons and synaptic terminals, and contains specialized organelles such as mitochondria, Golgi apparatus, endoplasmic reticulum, secretory granules, ribosomes and polysomes. The amount of the cell organelles varies with the functional activity of the cell. Delicate cytoplasmic threads called neurofibrils are there throughout the entire length of the axon and dendrites arising from the cell body. The cell body and its processes are surrounded externally by a thin membrane called the neuron membrane. The cell body is present in gray matter of the central nervous system of the brain and spinal cord. The third one is neuronal membrane. The neuronal membrane serves as a barrier to surround the neoplasm inside the neurons and to exclude various substances. The membrane is made up of lipids and proteins, fats and chains of amino acids. The basic structure of neuronal membrane is a bilayer or sandwich of phospholipid. The external side of the membrane have the receptors for some molecules. Whenever a molecule attaches to receptors, some changes of the membrane and in the interior of the cell ensure, such as alteration of the permeability to some ions. In this figure you can see the anatomy of a multipolar neuron. Next part is axon. The axons are long cytoplasmic process of the cell body which transmits impulse from soma to other neurons. The axon arises from the cell body in a conical elevation called axon hillock. The length of the axon is variable and depends on the functional relationship of the neuron. The axon with its surrounded sheet is a nerve fiber. The nerve fibers or axons are wrapped in myelin sheet. The region where the neuron is not covered by myelin sheet is the joint of neighboring myelinated parts is known as node of Ranvier. The axon is main conducting unit of the neuron. It is capable of conveying electrical signals along distances that ranges from as short as long as 1 meter. The next are nerve endings, or these are also called as presynaptic terminals. Axon give rise to branches termed collaterals along its course, and near the end it undergoes substantial branching into axon terminals or end brush, the last part of which is enlarged to form an end bulb. Types of neurons. Neurons can be of many types. Most neurons can be anatomically characterized as unipolar neuron or pseudo-unipolar. In these, dendrite and axon emerging from the same process. The examples are touch and pain sensory neurons. The second one is bipolar neuron. In these neurons, axon and single dendrite on opposite ends of the soma. The examples of bipolar neurons are retinal or olfactory neurons. The next category is multipolar neurons. In these, 2 or more dendrites separates from the axon. The examples are motor, pyramidal cells, Purkinje cells, or granular cells. The next one is anexotic neurons, where axon cannot be distinguished from dendrites. Unipolar neurons are unusual as they do not have dendrites. However, they still relay a signal from one cell to another. Although there is always only one axon, but it can branch out before it reaches its target. This figure is showing different kinds of neuron. For the first one is unipolar neurons, the second one is bipolar, third figure is showing multipolar neurons, and fourth one is pseudo-unipolar neuron. Now coming to the conduction of nerve signals. Nerve signals or impulses pass through neurons in the form of electrical signals. These signals cross the synapse. These synapses are nothing but small gaps between one neuron and the following neuron in the chemical form before being transmitted again in electrical form. Signals are also chemically passed onto the other targets like muscles, which make proper responses. Synapses can be electrical or chemical. Electrical and chemical signals. As an electrical signal arrives at the end of a fiber, it activates the discharge of neurotransmitter, which then sends the signal in chemical form to next neuron. Neurotransmitters are the brain chemicals that pass the information throughout the brain and the body and also relay signals between neurons. There are two types of neurotransmitters, excitatory and inhibitory. They are made in the neuronal cell body and then transferred to the axonal terminals or nerve endings. Each nerve ending is coupled to another neurons by a synapse. The physical and chemical nature of each synapse determines the strength and polarity of a new input signal. At this place, the brain is most flexible and mainly vulnerable. Synapses. The synapse contains a tiny gap separating neurons. Synapses are junctions formed with other nerve cells where the presynaptic terminal of one cell comes in contact with postsynaptic membrane of another. At these synapses, neurons are excited, inhibited, or modulated. Two types of synapses are there, electrical and chemical. An electrical synapse is a mechanical and electrically conductive link between two adjacent neurons that is formed at a tiny gap between the pre- and postsynaptic neurons known as gap junctions. Electrical synapses occur where the presynaptic terminal is in electrical continuity with the postsynaptic neuron. Electrical synapses conduct nerve impulses faster compared to chemical synapses and are often found in neural systems that require the fastest possible response. Chemical synapse. Chemical synaptic junction is more complex. The gap between the post and presynaptic terminal is big and the mode of transmission is not electrical but carried by neurotransmitters discharged at the presynaptic area of the junction. There are two types of chemical synapses. The first one, it is an excitatory synapse commonly found on dendrites, and the next one is inhibitory synapse. It is generally found on the cell bodies. Different substances are released at those two types of synapses. The direction of flow of information is typically one way at these junctions. In this structure, you can see a structure of a typical chemical synapse where the presynaptic axonal or nerve terminals and the postsynaptic cell membrane. Now coming to the neuronal communication. Neurons communicate by sending electrical signals. These are called action potentials. Action potentials are produced by the movement of ions across the neuronal membrane. Neurons carry information or messages in the form of electrical signals. These are called nerve impulses. Neurons have to be excited to create a nerve impulse. different stimuli such as sound, light, or pressure all excite neurons. But in most cases, neurotransmitter released by the other neurons will generate a nerve impulse. And when a nerve impulse reaches the end of one neuron, a neurotransmitter is released and it diffuses from the neurons across the junction and excites the next neuron. And the activation of membrane receptor either result in depolarization, that is an excitatory postsynaptic potential, makes it more likely that an action potential will fire, or leads to hyperpolarization, it is an inhibitory postsynaptic potential, it makes less likely that action potential will fire. This is showing the action potential. There are different states or phases of action potential. First one is the resting state. In resting state, the membrane, it's in resting state. The value of the membrane potential is about minus 70 millivolts, and then it is followed by depolarization. Depolarization means membrane becomes more positive. After that, there is a rising phase of action potential. After that, the action potential reaches peak and comes the falling phase of action potential. After falling phase, it goes undershoot. Ion movements and the action potential. The whole basis of action potential is based around a change in permeability to sodium and potassium due to opening or closing of voltage-gated ion channels. Two discrete states, open or conducting state and closed or non-conducting states. Some channels have also inactivated states, that is nothing but open non-conducting state. The action potential is all or none process. Either it is not triggered at all, example too little depolarization, or membrane is refractory, or it is triggered completely. Course of action potential. The first one is rapid depolarization. When partial depolarization reaches the activation threshold, voltage-gated sodium channels open and the sodium ions rush in. The membrane potential reaches to plus 40 millivolts from minus 70 millivolts. Then repolarization. Sodium channels close and become refractory. Depolarization triggers opening of voltage-gated potassium channels. Potassium ion rush out of the cell, repolarizing and then hyperpolarizing the membrane. That time you saw the undershoot, that was hyperpolarization. Next is resuming the resting membrane potential. During that, potassium channel close, repolarization resets sodium ions channels. Ions diffuse away from the area. Sodium-potassium transporter maintain polarization and the membrane is now ready to fire again. Essentials in chemical synaptic transmission. Action potential travels the length of axon of a neuron. When the action potential reaches the presynaptic terminal, it stimulates the release of a small quantity of neurotransmitter which binds to receptor situated in the membrane of another postsynaptic neuron on the opposite side of the synaptic cleft. This figure is showing typical mammalian neurons. Different types are multipolar interneurons, a motor neuron, and a sensory neurons. The arrow indicates the direction of conduction of action potential in the axons. How neurotransmitters work. The job of neurotransmitters is to carry nerve impulses across the synapse between neurons and target cells. Neurotransmitters either stimulate or inhibit electrical impulses in the target cells. The arrival of a nerve impulse stimulates the discharge of neurotransmitters from presynaptic vesicles. Then they pass across the synapse and open channels in the target cells on postsynaptic neuron. Charged particles then enter and trigger the second impulse. Blood-brain barrier or BBB. And structure and function of this blood-brain barrier. This is a permeability barrier that separates brain from the circulatory system. It protects the central nervous system from many substances like viruses, bacteria, and harmful chemicals, while regulating the transport of necessary substances. The blood-brain barrier is formed by a monolayer of specialized endothelial cells which are connected by tight junctions and transduce signals from the vascular system and from the brain. Astrocytes, these are essential to create and contribute to the differentiation of blood-brain barrier. Brain capillaries form a tight barrier except in specialized areas. The structure and function of the blood-brain barrier is dependent upon the complex interaction between different types of cells such as endothelial cells, astrocytes, and the pericytes, and also extracellular matrix of the brain and blood flow in the capillaries. Anatomy of blood-brain barrier. The blood-brain barrier is high-density cells lining limiting the passage of substances from the bloodstream much more than do the endothelial cells in the capillaries at any other place in the body. Astrocyte cell projections called astrocytic endfeets enclose the endothelial cells of blood-brain barrier, giving biochemical support to these cells. In this figure, you can see the detailed structure of blood-brain barrier. First view is the brain, and next is you can see the capillary, astrocytes, and the pericytes. And the last one, more details endothelial cell layer is visible. Non-neuronal or supporting cells in the brain. These cells are called glial cells or simply glia. These are quite different from neurons. They give support and protection to the nerve cells, but they do not transmit nerve impulses by themselves. The glial cells are non-neuronal cells that form myelin. They surround neurons and hold them in position, maintain homeostasis, and provide support and protection for neurons in the central and peripheral nervous system. There are different types of neuroglia in the central nervous system and the peripheral nervous systems. This figure is showing both types of glial cells. Types of glial cells. The first one is microglia. Microglia are the specialized macrophages. These are capable of phagocytosis that protect neurons of the central nervous system. These cells are found in all brain areas and spinal cord. Microglial cells are relatively smaller than macroglial cells with changing shapes and oblong nuclei. They are moved within the brain and multiply when the brain is injured. Coming to the second one, macroglia. They derive from the ectodermal tissue and they are of different types. First one is astrocyte, or these cells are also called as astroglial cells. These are the most abundant cell types of microglial and are found throughout the central nervous systems. An astrocyte is an starch-like cell and has many processes extended from its cell body into the surrounding networks of the nerve fibers. The next are oligodendrocytes. These are the cells that trap the axon in central nervous system with their membranes forming a myelin sheet. The myelin sheet provides insulation to an axon that permit electrical signals to spread more efficiently. Oligodendrocytes have fewer and thinner processes than the astrocytes and no gap junctions are there. Ependymal cells are found in central nervous system. These cells are involved in the formation and secretion of cerebral spinal fluid, and they beat their cilia to help circulate the CSF and make up the blood cerebrospinal fluid barriers. Next is radial glia. These cells arise from neuroepithelial cells after the onset of neurogenesis. Schwann cells, they are found in peripheral nervous systems, that is around the nerve of extremities of the body, like in the locations in the skin. Schwann cells provide myelination to the axons in peripheral nervous systems. Satellite glia, these cells are small that trap neurons in sensory, sympathetic and parasympathetic ganglia. Anterior glial cells are found in intrinsic ganglia of digestive system. Transport across the blood-brain barrier. The blood-brain barrier is highly selective permeability barrier that separates the circulating blood from the brain extracellular fluid in the CNS. The blood-brain barrier has a number of highly selective mechanisms for transport of nutrients into the brain. Transport across the blood-brain barrier may be passive or active. The passive transport, there is no energy required to pass. For example, some uncharged molecules like oxygen, carbon dioxide, hormones, molecules that can dissolve in the fats of the capillary walls. For active transport, energy is required to pass. Example, glucose, amino acids, vitamin, etc. In this diagram, it shows several ways in which transport across blood-brain barrier works. So students, let us now summarize what we have learned in this module. So we have learned so far that nervous tissue consists of two main types of cells. These are neurons and neuroglia. And neurons are the nerve cells that originate, process, transmit and receive nerve impulses. And neurons transmit nerve impulses that are sent information around the body. Nerve impulses travel electrically along the nerve or neuronal membrane and are transmitted by chemical messenger, and these messengers are called neurotransmitter, to the next neuron across a tiny gap called synapse. In addition to neurons, the nervous system contains large number of other cell types. These are called neuroglia, and these neuroglia support, protect and nourish neurons. And glia have many functions in support of nerve cells, but they do not transmit nerve impulses by themselves.